next topic is gene therapy. Now, partially we have already studied about gene therapy when we discussed about recombinant DNA technology. Recombinant DNA technology is nothing but gene therapy. Alright. Now, this gene therapy also can be very useful in the medical procedures. What do we do in gene therapy? Remember, there are three things. We had discussed about this. I am writing this again. Either addition or removal or modification. Correct? These are the three things that can be done. So precisely in gene therapy, this is what we try to do. That if, let's say, for any medical therapy, gene therapy is being used, then either what we'll try to do is we'll locate the diseased gene, we'll take it out of the system, or we will locate the diseased gene, we'll replace it with a healthy gene. Or what may happen is that certain disease has happened in an individual because of absence of a given gene. So if there is an absence of that gene, we will try to add that gene artificially. So in this case, either addition or removal or modification. All these three become a part of gene therapy. So gene therapy is a medical technique that manipulates genes to treat or prevent a disease. The gene therapy is focused on the following approaches: either replacing a gene responsible for disease with a healthy gene, or inactivating or knocking out a gene that is functioning improperly, or introducing a new gene into the body to help fight a disease. So that's why addition, removal, or modification, all these three are possible. So that's why gene therapy can be used as a treatment option for a number of diseases. For example, inherited disorders, that is, we are talking about genetic diseases or the genetic disorders, some types of cancer, and certain viral infection. So against these, we can make the use of gene therapy. But the problem with gene therapy is that we haven't seen gene therapy to be very successful. First of all, we have seen that the nature of the treatment is short-lived. Now what happens is, let's say you introduce a gene, correct? Now what we have discussed is that our cells will keep on dying and they will be replaced with new cells all the time. So there is rapid multiplication of cells that is happening in our body. So many a times what happens is that initially when gene therapy has been done, it is functional for some time, but over a period of time, the body starts to behave back in a similar manner as it was behaving earlier. So that's why this gene therapy that was working for some time, after many cycles of cell division, ultimately it may not be functional anymore, and this would have been replaced by the old system of working in the body. So that's why there is a possibility that this gene therapy will be short-lived. This is one reason why gene therapy hasn't been seen to be very successful. Then after that, immune response. Now just know this, that why do, for example, vaccines work for us? Vaccines work for us mainly because our body sees even the vaccine as something which is foreign. So that's why it starts to act against this vaccine, and it starts to produce a lot of antibodies against this vaccine. And ultimately these antibodies only help us if at all the same disease against which the vaccine has been given to us sometime or any point in time visits our body. Alright? But ultimately what is happening? Our body sees even the vaccine as something which is foreign, and for the immune system, the funda is very clear that if there is something which is foreign, you have to attack it and you have to kill it. This is something that also even happens with gene therapy. That if gene therapy is being carried out, let's say there is a new additional gene added to your system. So because it is additional, it has come from outside, it is a foreigner for our immune system. Our immune system may act against this gene, and many a times it may be able to knock out the gene or destroy the gene. So ultimately the gene therapy does not work in such a case. So what may happen is that the gene therapy might activate the response of our immune system. Even our immune system reduces the effectiveness of gene therapy. Alright, so this is another problem that may happen. Apart from that, what happens is that many of the disorders are multi-gene disorders. Meaning what? It means that there are multiple genes being affected at the same time. So now the gene therapy now has to be carried out against multiple genes, which becomes very complex as a process and becomes very difficult to implement. So that's why in multi-gene disorders, it is highly unlikely that gene therapy would work. So that's why if you talk about, for example, high blood pressure, heart disease, Alzheimer's, arthritis, diabetes, all these are multi-gene disorders. All right, all these are multi-gene disorders, and that's why in these kind of cases, using gene therapy becomes equally more difficult, and probably it will not be as effective. So because of these three reasons: one, the short-lived nature of gene therapy; second, our immune response; and third, some of the disorders being multi-gene disorders, we see that gene therapy is not very effective generally. There are only certain cases where gene therapy has been seen to be very effective. In most of the cases, it hasn't been seen to be that effective. So that's why currently we see that gene therapy is currently being tested only for treatment of diseases that have no other choice. All right. So just remember this. The next topic is the topic of three parents baby. And this is a concept which has taken a lot of center stage in perhaps the last decade, especially in the last 6-7 years. What is this concept? Why are we calling this as three parents baby? Now, what happens sometimes is that there could be some problem with the mitochondria of the mother. And if mitochondria of the mother is defective, then the same mitochondria will be transferred to the child or the offspring as well. And this is precisely where the concept of three parents baby comes into the picture. Now, when we talk about the three parents baby concept, what we do in this case is very simple. That we take the mother's egg cell. From there, we take out the nucleus. All right, we take out the nucleus. As you can see, the nucleus has been taken out. Then there is a donor egg cell. And in this donor egg cell, first of all, we will remove the nucleus from the donor egg cell. And this nucleus from the mother will be taken and it will be inserted in the donor cell. Now, there are two things. One, that why are we doing this with the nucleus? Why are we replacing the nucleus only? One. And second, if the problem was with mitochondria, why to replace the nucleus? Why are we not replacing the mitochondria itself? Right? Let's address the first problem. Why are we replacing the nucleus? What is special about the nucleus? Remember when we discussed about cloning? In case of cloning also we had discussed something, that nucleus has all the characteristics of an individual. Nucleus, inside the nucleus when you go, you will find that all the genes are present inside the nucleus. All the DNA are present inside the nucleus. And these DNA make up the genes. So these genes which are specific to a person, they are inside the nucleus. So that's why nucleus becomes very, very important. And if you want a child to have the same characteristics as the mother, then in that case you need to make sure that the nucleus is being taken from the original mother. Otherwise, the characteristics or the genetic makeup of the child will come from the donor mother. It will not come from the original mother. So that is the reason why we take out the nucleus from the original mother. Now, the second question is, if the problem is with mitochondria, why are we taking out the nucleus? The reason behind this is that mitochondria are present in large numbers in the animal cell, or even, for example, in the human cell, which is a kind of animal cell. So in the animal cell, you have large number of mitochondria, but you have one single nucleus. All right, you have one single nucleus and many mitochondria. So that is the reason that why nucleus is taken out and not the mitochondria. Because just imagine that instead of picking out just one nucleus, if you had to pick out 20 mitochondria, it would be a much more difficult task. Correct? So that's why we don't take out the mitochondria. We let the mitochondria be there, but we just take out the nucleus. So we take out the nucleus from the original mother. This nucleus will have all the characteristics of this mother, this particular person, and we take away the nucleus. We remove the nucleus from the donor egg cell. We remove the nucleus from the donor egg cell because this nucleus would have had all the particular characteristics of the donor. So that's why the enucleated cell that we have, enucleated egg cell that we have, it will not have the characteristics of the mother. And then when you place the nucleus inside the donor, this is where the egg cell with original mother's nucleus is ready, and this will now fertilize with the sperm cell. So fertilization will still happen between the nucleus of the original mother and the nucleus of the father. All right. Donor, what is the donor doing? The donor is providing the mitochondria, healthy mitochondria. Now, what is the importance of providing mitochondria? And what we are saying is, just remember this. I just hope that you noticed that what we said, what we started with was that the genetic diseases in the mitochondria will be transferred to the child. Correct? The genetics or the mitochondria are defective, and the same mitochondria, the genes of these mitochondria will be shifted to the child. All right. So what does it mean? It also means that mitochondria also has genes. Now this is another question that has come in the past, way back. I think it was 2004 prelims examination when this question was asked. There were three options given. One was nucleus, second was mitochondria, and if I'm not wrong, I could be wrong here, the third one either was chloroplast or either they had asked about plastids in general. And the question said, which of the following contain DNA? Which of the above contain DNA? Alright, and then you had the four options: 1 only, 1, 2 and 3, 1 and 2, 2 and 3, etc. The answer actually was 1, 2 and 3. So what you should remember is that nucleus does contain genes, but at the same time mitochondria also contains genes. But there is a difference between the genes of mitochondria and the genes of the nucleus. What is the difference? The difference is that the nucleus contains specific properties of that individual, and mitochondria contain general properties of all the individuals of a particular species. What do I mean? Let's say if I talk about my nucleus, all right? So the genes that are there in my nucleus, they would have specific characteristics of me. How I look, what is the shape of my face, what is the amount of hair on my scalp, what is my height, what is my skin color. These are specific to me, correct? These are the things which are specific to me. So these kind of characteristics are stored inside the genes of the nucleus. All right. These kind of things which are particular to an individual, these kind of characteristics are there inside the nucleus. All right. The genes of the nucleus. What does the gene of a mitochondria have? The genes of mitochondria, they contain normal or general characteristics. What are general characteristics? How does the heart beat? How do I breathe? Correct? How do I walk? So walking, breathing, beating of the heart, response of the brain, these are the things which are general in nature, correct? For every individual, it's the same. For every individual of a particular species, it's the same, right? So that's why mitochondria contain the genes which are non-specific, which have characteristics which are non-specific, which are general among that species. But nucleus contains the characteristics which are specific to a given individual. So that's why we want the nucleus. That is the reason why we need the nucleus of the original mother, because then the child who will be born, the child will have all the characteristics of original mother as well as original father. That's why in the UK back in 2015-16, there was a case that went on, that in this kind of a concept of a 3-parent baby concept, can the donor mother also have a claim over the child? The answer was no. The ruling of the courts were no. Why? Because it simply said that, look, all the characteristics are coming from mother and father. The only thing that the donor gave was mitochondria, and those mitochondria could have come from anyone. It could have come from thousands of other mothers, but the specific characteristics of the nucleus of the original mother could not have been given by anybody else. So that's why the claim over the child born out of this kind of a procedure will still lie with the original mother and the original father. This is the ruling that was given by the courts back in 2015 and 16. So, three parents baby, human offspring produced from the genetic material of one man and two women through the use of assisted reproductive technologies, specifically mitochondrial manipulation or replacement technologies, and 3% in vitro fertilization. In other words, it is also called as pro-nuclear transfer. Pro-nuclear transfer is another name that is given apart from mitochondrial manipulation and 3% in vitro fertilization. These are other names which are used for the concept of three parents baby. Alright. There's one question that had come in the examination. In the context of recent advancements in human reproductive technology, pro-nuclear transfer is used for A. Fertilization of egg in vitro by the donor sperm, genetic modification of sperm producing cells, development of stem cells into functional embryos, prevention of mitochondrial diseases in offspring. This is what we are doing, correct? What are we doing in pro-nuclear transfer? What is the other name? Three-parent baby. We just had discussed about this. So that's why the correct answer here will be D. A direct question was asked from me.